Have you ever wondered how scientists can study the tiny powerhouses of our cells like mitochondria or the cellular brain, the nucleus, all by themselves? It seems impossible, right? Like trying to separate the ingredients of a cake after it's been baked. But in biology, there's a clever technique for just that, and it's called cell fractionation. Today we're going to dive deep into this process and see how it unlocks the secrets hidden inside every cell. Stay tuned because this is where biology gets really cool. Cell fractionation is a fundamental method used in cell biology to separate different parts of a cell, known as organelles, so they can be studied individually. Think of it as a microscopic sorting facility. The whole point is to isolate specific components like the nucleus, mitochondria, or ribosomes from a cell. This is crucial because to understand what a single part does, you need to study it in isolation away from the bustle of the entire cell. This process relies on two key physical properties, size and density. By separating organelles based on these differences, scientists can get a pure sample and analyze its structure using tools like the electron microscope. The entire process can be broken down into two major stages: homogenization and centrifugation. Let's start with the first one. The first step is homogenization, which literally means to make uniform. The goal is to gently break open cell membranes to release organelles into a solution, creating a soupy mixture called a homogenate. Imagine putting a bunch of fruits into a blender to make a smoothie. It's a similar idea, but way more delicate. Scientists use special equipment like a mechanical homogenizer or even high-frequency sound waves in a process called sonication. The cells are suspended in a special buffer solution. This isn't just any liquid. It's carefully prepared to mimic the cell's natural environment. It's kept cold to prevent enzymes from digesting organelles, has a specific pH, and contains sugars to prevent them from bursting. For plant cells, which have a tough cell wall, enzymes like pectinase and cellulase are often added to digest the wall. Once we have this uniform suspension of cellular parts, we're ready for the main sorting event, centrifugation. Now for the second and most exciting part, centrifugation. This is where the actual separation happens. The cell homogenate is spun at incredibly high speeds. This creates a powerful centrifugal force which is much stronger than gravity. Just like when you're on a fast spinning merry-go-round and you feel pushed to the outside, the same principle applies here. The key is that heavier, denser particles experience this force more strongly and move to the bottom of the tube faster than lighter ones. This allows us to separate organelles. The most common method used for initial separation is called differential centrifugation. Differential centrifugation means we do it in steps, with each step separating out different components. We start by spinning the homogenate at a low speed. The largest organelles, like the nuclei, form a solid clump called a pellet. The rest of the liquid, which still contains smaller organelles, is called the supernatant. We carefully pour off this supernatant into a new tube and leave the pellet of nuclei behind. Now we take the new tube and spin it again, but this time at a much higher speed. This increased force pulls down the next heaviest organelles, typically the mitochondria and, in plant cells, chloroplasts. We can repeat this process several times, each time increasing the spinning speed. After mitochondria, we can pellet cell membranes and finally, at very high speeds, the tiniest particles, the ribosomes. So through this step-by-step process, scientists can effectively build a library of isolated cell parts. These purified samples are now ready for experimentation. Scientists can study reactions that happen only in specific organelles. It's this ability to deconstruct the cell that has allowed us to understand how life works at the molecular level. From the role of mitochondria in energy production to how DNA is organized, cell fractionation has been at the heart of breakthroughs. And there you have it, the elegant and powerful technique of cell fractionation. It's a perfect example of how physics and biology work together to help us explore a world invisible to the naked eye. By breaking cells apart, we can piece together the intricate puzzle of life, one organelle at a time. I hope you found this journey into the cell fascinating. Please give it a thumbs up and subscribe for more deep dives. Thanks for watching.